All right, let's jump right into this explainer. Today we're tackling a massive, highly tested concept for your medical board exams, the clinical implications of cellular mechanisms. We're going to look at the exact molecular switches that tell a cell to divide, and more importantly, what happens when those switches completely break down.
It's honestly one of the most fundamental questions in medicine, right? Why do some damaged tissues bounce back flawlessly while others spiral into life-threatening tumors? Well, it all boils down to the microscopic security systems guarding the cell cycle and what happens when they fail. Understanding these mechanisms is the absolute bedrock of modern oncology and pathology.
Here's our roadmap for today. We're covering mitotic activity and tissue types, the phases of the cell cycle, cyclins, kinases, and checkpoints. Then we'll hit mitosis and the anaphase complex, dive into the pathology of P53 and cancer, and wrap it all up with a high-yield board review.
All right, Section 1, mitotic activity and tissue types. Let's look at how mitotic frequency actually dictates tissue regeneration.
This is a classic exam contrast. Normal growth heavily depends on the timing of cell division, and this timing, it varies wildly. Think about your skin. Those epithelial cells are constantly turning over. They're a labile population, regenerating continuously, usually on a tight 12 to 24-hour cycle. But then on the total opposite end of the spectrum, we've got permanent populations. Think highly differentiated cells, mature nerve cells, heart muscle cells. Once these guys reach maturity, that's it. They are permanently locked out of the division cycle. And sitting right in the middle, we have stable cell populations. These are super interesting because they actually keep their regenerative abilities in reserve. Take your liver cells, for example. They rarely divide, maybe once every year or two, and they mostly just hang out in a resting phase we call G0. But if the liver gets damaged, these cells can absolutely be called upon to step up, re-enter the cycle, and divide.
Moving on to Section 2, phases of the cell cycle. So what's actually happening under the hood when a cell commits to dividing? Just think about the sheer scale of the phases here. A massive proportion of the cycle is dedicated to interphase. That's G1, S, and G2. Compare that to the super brief, roughly 1-hour window of the actual M phase, or mitosis. It's crazy. A cell spends the vast majority of its life simply preparing to divide.
For your exams, you absolutely have to memorize these key events. In G1, the cell is synthesizing enzymes, getting everything ready. But the S phase, that's where the magic happens. DNA replication kicks in, and all those chromosomes are duplicated into sister chromatids. Now pay close attention to G2. This phase is all about high microtubule production. The cell is literally manufacturing the structural cables it's going to need to pull itself apart. Clinically, this is exactly where certain chemotherapeutic agents strike. They interfere with that microtubule production to halt cancer in its tracks. Oh, and remember that G0 resting phase we just talked about? It's not just a waiting room for stable cells, it can also be a quarantine zone for damaged cells. Basically, it acts as an alternative to programmed cell death.
Section 3, cyclins, kinases, and checkpoints. Okay, so we know the phases, but how exactly does the cell know when it's time to move from one to the next? I want you to think of these internal controls like a system of strict biological traffic lights. The engines running the system are cyclin-dependent kinases, or CDKs. Now here's the crucial point. While these CDK enzymes are always hanging around in the cell, their overall numbers don't really fluctuate, they are totally useless, completely inactive. That is until they get bound by their specific partner proteins, called cyclins. As the concentration of different cyclins rises and falls throughout the cycle, they bind to those CDKs. Once this complex forms, boom, the CDK is activated. It acts like a master molecular switch. It starts adding phosphate groups to other protein substrates, which essentially forces the cell to execute specific events and push past critical restriction points. And once the cell safely moves to the next stage, those active cyclins get degraded.
These cyclin CDK complexes are literally what power the cell through its checkpoints. The G1 checkpoint is asking, hey, are we ready for DNA replication? Then the G2 checkpoint acts as a super strict quality control stop, checking for any DNA damage before allowing division. Finally, the M checkpoint ensures all the spindle fibers are properly connected. If conditions aren't absolutely perfect at any of these stops, the whole process gets assessed and halted. All right, Section 4, mitosis and the anaphase complex. Let's assume our cell successfully passes that stringent G2 checkpoint. Now we enter the dramatic, roughly 1-hour process of karyokinesis, or nuclear division. The classic sequence of PMAT, prophase, metaphase, anaphase, and telophase, ensures that those sister chromatids we built back in the S phase are perfectly segregated. By the way, this mechanical process is also vital during gametogenesis in meiosis. Basically, the nuclear envelope breaks down, the chromosomes line up right at the equator, and then they are forcefully ripped apart to opposite poles. When you visually compare the beginning and the end of mitosis, you can see exactly how those microtubules we manufactured in G2 do their heavy lifting. In prophase, the spindle fibers are just starting to emerge. Fast forward to telophase, and those spindles have successfully dragged the genetic material to completely opposite sides of the cell. Then, the nuclear envelope begins to rebuild itself around the two new identical sets of DNA. To really nail the clinical implications here, you need to understand exactly how they separate. At the M checkpoint, the cell double-checks that every single chromosome is perfectly aligned. Once it gets the green light, it activates the anaphase promoting complex, or APC. Think of the APC as a destroyer. It initiates the destruction of cohesin proteins, the literal glue binding the sister chromatids together, and it decomposes M-cyclin. Without that cohesin, the chromatids are finally free to be pulled apart. Section 5. Pathology, P53, and cancer. So what happens when this perfectly orchestrated system of checkpoints completely breaks down? Clinically speaking, this breakdown is the origin of tumors. Cancer isn't some alien invader, right? It's essentially a massive failure of the cell's very own division controls. When those external and internal controls are lost, you get unrestrained, uncontrolled cell growth. And in patient diagnostics, these specific cycle malfunctions actually serve as vital tumor markers for oncology. And that brings us to arguably the most important molecule in all of oncology, the tumor suppressor gene P53. We like to call it the cell cycle enforcer. Normally, if DNA damage happens, say from heat, radiation, or chemicals, P53 detects it right at the G1/restriction point. It throws on the brakes, halts division, and either stimulates repair enzymes or forces the cell into G0. If the damage is totally beyond repair, P53 triggers the destruction of the cell. But if P53 is abnormal, which we see in clinical case studies of certain really aggressive tumors, the cell completely ignores the damaged DNA. It just keeps dividing and dividing, accumulating more mutations until it turns cancerous. In fact, literally all cancers must shut down P53 activity in order to bypass this restriction point. When normal P53 realizes a cell simply can't be fixed, it triggers apoptosis. Now, apoptosis is MEAT, controlled cellular suicide. The key enzymes here are caspases, and they act like molecular scissors. The cell condenses, the organelles remain intact, and crucially, there's absolutely zero inflammatory process in the adjacent tissues. Contrast that with necrosis. Necrosis is chaotic cell death, caused by severe external damage like hypoxia or extreme temperatures. The cell completely loses control of its fluid balance, swells up, and literally breaks open, causing massive, damaging inflammation in the surrounding tissue. A major clinical challenge we face today is when cancer cells develop drug resistance by mutating to bypass those normal apoptosis pathways, essentially keeping themselves artificially alive. All right, home stretch, Section 6, High-Yield Board Review. Let's rapidly synthesize the absolute most critical takeaways you're going to need for your exams. Let's do a rapid-fire review of concepts you'll definitely want to remember. Board examiners love, and I mean love, to test your knowledge of how external and internal factors push and pull the cell cycle. Mitogens? Those are your accelerators. They bind to surface receptors and override negative controls to force cell division. Survival factors act like shields, directly suppressing the apoptosis pathways so the cell stays alive. The APC is your separator, destroying cohesin. And finally, P53 is your master regulator, the ultimate brake pedal. So I want to leave you with this provocative thought. We know that bypassing the P53 enforcer is essentially required for cancer to thrive, allowing it to ignore DNA damage and replicate wildly. But if we can map the precise cyclins and kinases that power the cycle forward, how could novel drugs designed to artificially block specific cyclin-dependent kinases change the entire future of oncology? Understanding these microscopic traffic lights isn't just about passing a medical board exam. It's the very foundation for developing targeted clinical therapies that could intercept and stop cancer right in its tracks. Thank you so much for joining me on this explainer, and keep studying those high-yield concepts.